Good morning. I am Dr. Vilasrao Gorpade, SGM College, Karad. Today we will discuss about B cell activation in the antibody production. Now, there are two types of the defense mechanisms are there. One is cell-mediated type of immune mechanism, and second one is antibody-mediated mechanism. So, resistance is quite different from the defense mechanism. Now, how the antibody production takes place, this is very important in the immune system. This is one, how these B cells are produced, and B cell and mature, once they mature, they produce the antibodies. And the antibodies play the important role in the defense mechanism, that is a antibody dependence phase, that is called as the antibody dependence phase. And once the multiplication of B cell starts, it will convert it into the plasma cell, and the plasma cell is secrete the antibodies, and the peripheral lymphocytic organ such as a lymph plays the important role in the defense mechanism. Now, there are two types of the antigen. One is called as antigen thymus dependent antigen, and second is called as a thymus independent antigen. Thymus dependent antigens are there. They are said to be a proteins 1 because they depend upon the helper T cells. And second one is independent antigen, thymus independent antigen. They does not require the T cell, or the helper cell is not required here, and they are majority of they are polysaccharide and the lipids. Now we will study what is the B cell response, the thymus dependent antigen, how it works and how antibody response is given. So there are two types of the immune response is there. One is called as a primary immune response, and second one is called as a secondary immune response. Now this is the best example of primary immune. Once the man is infected first time in the life with any bacterial antigen, that is called as a primary immune response. And when second time he is going to infect with the same one, then it is called as a secondary immune response. And this primary immune response is having the primary infection, and there is a production of the antibodies. That majority of the antibodies are IgM and the IgG type. And in case of secondary infection, when the same antigen is going to infect the person, it is called as a secondary antibody response, whether the IgG is the major antibody is produced in the secondary immune response. So this is primary immune response is last for 5 to 10 days, and secondary immune response is last for usually 1 to 3 days. And the peak response, it is a primary immune response is very small, while in secondary immune response it is very large. The antibody isotypes is generally IgG, and the IgM is always greater than the IgG, but here is the IgG is the major immunoglobulin is synthesized along with the IgA and IgE. Lower average affinity is more variable, where the higher average affinity we will find in the secondary immune response. Now, how the humoral response is shown? Humoral response means antibody mediated response. See, this is the example of this, how the antigen recognition is carried out, how the activation of B lymphocyte takes place, and once activation of B lymphocyte takes place, how the maturation and the memory cell is kept. That is the example. So, when this is a microbes are there, these microbes are there, and these microbes are attached to the B cell. This is a native cell. They recognize the antigen, and this when binding of microbes takes place to the B cell, there is an activation of the B cells are there with the help of T cell stimuli. I told you that helper cells are very important, plays the important role. They sensitize to production of the antibodies. When these activated B cells are there, that activation takes place, there will be a multiplication and the proliferation of the takes place, and the clonal expansion takes place. This is a clonal expansion is there. Once this activation takes place, the clonal expansion is there. Once this clonal expansion takes place, differentiation of these cells again takes place. And the effector is carried out, effector cell antibody secreting cells are there. These effector cells secrete the antibody secretion is takes place. This is IgM. And second thing, this is a class switching, that is this IgM is shifted to the IgG. And sometimes or all the times the high affinity expressing B cell, they produces the memory cells, and the memory cell is kept for a longer period of time. Because when you are suffering from the second type of infection, there will be immediately peak period will be happen. Now this antigen doesn't take part in the chemical reaction or antibody-antigen reaction. There are certain parts on the antigen that takes part in the antigen-antibody reaction mechanism. This is one that is called as epitopes. You might have studied that epitopes are active sites in the enzyme. There is no difference between active sites and the epitopes which is present on the antigen. So antigen which contains of the epitopes, these epitopes takes the part in the chemical reaction. They play the important role they play in case of antigen-antibody reaction. Whole the bacteria doesn't take part in the antigen-antibody reaction. So this is called as a epitopes. Now, there are major histocompatibility complex. There are two types of the complex are available. One complex is a major histocompatibility complex 1 and major histocompatibility complex 2. Now what is this? There is both are plays the important role in the defense mechanism. And the MHC1, the most of the target cell can present the antibody, antigen, and nearly it is a nucleated cell infected by microorganism or the viruses. Abnormal protein is produced by the cancer cell, aging of the cell by allogenic cell from the transport, and associated W and MSC1 require the replication of the foreign entity. What is foreign entity? There should be replication of the antigen should be there to reach the target site. While if you see MSC, major histocompatibility complex 2, that only APC can present the antigen. What is the APC? That is antigen presenting cells speaker. There are two types of the speakers. One is professional APCs and second one is non-professional APCs. They are associated that MSC does not require any replication of the entity. This is the basic difference. Here the replication is very important. Here replication doesn't have any importance in the target cells. So here in MSCC, which present the phagocytosis of the antigen processing, the phagocytosis is carried out. Now, how this antigen is processed through the separate pathway? There are two types of the pathways. One is called as cytosolic degradation and endocytosic degradation. And MSC1 interact with the cytosolic degradation, while MSC second interact with the peptide endocytosic degradation. Now see here, this is a cytosolic pathway. This endogenous antigen here, that is a cytoplasmic protease complex is there. They convert into the peptidase and it is reached to the endoplasmic reticulum, and this peptide class 1 react with the MSC complex. This happens in cytosolic pathway, only this one. Or endocytolic pathway, or the endocytic pathway, this exogenous antigen is there. This is endogenous antigen, this is exogenous antigen, and endocytosis or phagocytosis is carried out, where a endocytosic compartment is there, peptides are synthesized, and this peptide react with the MSC complex 2. So here MSC complex first, here MSC complex 2 is there. Now this processing of endogenous antigen, how it is takes place, a cellular protein are constantly regulated and most have the brief half-life, and it is turned over the same hole to the endogenous antigens. And the processing is carried in the three steps. First stage is a peptide generation, or it is called as a proteolysis. And second one, it is transported. Once this proteolysis is over, this peptide is taken towards to the endoplasmic reticulum, and then the peptides binds with the MHC complex 1. Now this is the best example and the diagrammatically represented. Just see that this is a campron protein. It is promoting folding, where these proteins are there, that is converted into the peptides, and these peptides are coming here, and they are binding with the MHC first, and then it is transported to this is endoplasmic reticulum. That is and it is exited through the endoplasmic reticulum, or it is with the help of plasma membrane. Now this is one assembly stabilization. How stabilization of measures histocompatibility 1 MHC 1 complex 1 antigen complex, how it is formed. That is a MHC1, that is a major histocompatibility complex bind with this one alpha chain. They are binding with together to and the peptides are there. Their peptides are joined together MHC first, and it exists with the help of RPR. Now what this endocytic pathway, exogenous antigen, how it is carried out with the help of endogenous antigen. This exogenous antigen, they are typically phagocytosis. Now you will have well known fact with the phagocytosis that the macrophages which carry out the phagocytosis. The foreign antigens are there, these foreign antigens are there, these foreign antigens are degraded by the phagocytic cell. That is, early endosomes is synthesized, that endosome is taken inside, endosome takes the antigen inside, then late endosome is synthesized, and afterwards that is converted into wholly degraded by the bacteria is degraded, and then it is called as a lysosomes. So this antigen which is degraded at 13-to-8 polypeptide which bind to the major histocompatibility complex second, and eventually endocytic vacuoles return to the PM and recycling to the surface receptor is carried out. Now this is an example of this, how it is carried out. This is a class of second, that is a major histocompatibility complex second bind with the urine chain, and this is a clip formation, that is a digestive inverted chain, that is a breakdown of the chain takes place and it is released to the peptide, and this is carried out. Now we will compare how this MHC feast, that is a endogenous activity, and exogenous activity, that is a exogenous pathway and endogenous pathway is carried out. In class Y, that is MHC, it is a endogenous, and in class 2 MHC, that is a exogenous. So this is the endogenous antigen is there, that is a proteasomes, it is coming inside, caliculum, endoplasmic reticulum is there. Here class 1 MHC is there with the Golgi complex, and there is a binding to the class 1 MHC, that is a major histocompatibility complex. It binds, the antigen binds to the major comma. Here this is exogenous source is coming inside, it is a phagocytosis here, and it is binding with the major histocompatibility complex second. Now this is the best picture of the lymph, how this lymph is there, medulla is there, pericortex is there, and the cortex. This is the entry of the antigen, how it takes place. Now how the activation of B cells takes place, this is very important. The activation when microbes are there, this microbe bind to the CD3D, that is a complement activation is carried out, and unless there is a complement activation, it should be recognized by the B cell, and once it is recognized by B cell, the signal will get to the Ig and CR2 complex, and once signal get to the CR2 complex, there is a B cell activation, and which starts the production of the antibodies. Now this antigen recognition is very important. See, almost all antigen cannot be recognized, that is very important. Recognition is very important. Unless there is a recognition, there will not be a reaction mechanism. And once there should be antigen antibody reaction mechanism should happen, then there should be a recognition is very important. So this is a lymph node. You will well know the fact, a thymus, lymph node, bursa fabricus, this plays the major role in the defense mechanism. Now this is a lymph node. This lymph node antigen presenting in the T cell division. So see, this is microbes are there. That microbes is APC adjacent to the APC. There are T cell proliferation and the differentiation is carried out. And T cells plays the important role in the cell mediated type of immunity. So there is effect of CD4 cells in the T cell, and this migrate towards this and binds to the interaction takes place of the antigen and the antibody. So in B cells, other lymphoid follicle cells, it is called as, that is called as a antibody mediated response. Whether these B cells are there, these microbes are attached to these antibodies are produced by the this B cell, and B cell activation is takes place, and B cell activation migration to the this one. So antigen recognition mechanism is the initial mechanism that activation should be very important. Unless there is activation, there is no proliferation of the antibodies. So this is our photograph of this one. This B cells is attached to these microbes. This is initial concept, and this is a conjugate is formed. Once this is said, that means this is an engulfment cell. So how the play that T cells, that is a helper cells are there, how helper cell helps in the recognition mechanism, how this helper cell in the cell mediated type of and defense mechanism. So these B cells are there, they present the antigen to the helper cells. So this is your antibodies, this is your B cells, this is CD40 cells, and this is a T cells. There is a cytokinin receptor is there. This cytokinin receptor is there. This cytokinin receptor, this helper T cell activate and express the CD40L. So CD1 ligand is expressed, there is secretion of the cytokinins. And once there is secretion of the cytokinin takes place, B cells are activated by engagement of the cytokinin, and there is a proliferation of the antibodies. So for production of the antibodies, the cytokinin plays the important role, and their cytokinin activation is carried out with the help of T cell, that is the helper cell. Now this is best example that the figure is there, how dependent of the antigen and antibody is carried out. Now there are cell dependent antibody response. How it is carried out in a germinal center reaction? How? First is affinity maturation is carried out. Then somatic hypermutation is carried out. Unless there is a hypermutation, there will not be a synthesis of the antibodies. So generation of memory B cells. Though what is this affinity hypermaturation and the affinity? What is a hypermaturation? The affinity maturation is the process which leads to increase the affinity of the antibodies to particular antigen in result of the somatic mutation, and the IgG gene followed by selective survival of B cell producing antibodies with the highest affinity. Means survival of the antibodies are very important to produce high amount of the antibodies. That is the survival, that the affinity should be there, and this affinity it is because of the somatic mutation which takes place in IgG. So these are the heavy chains and the light chain. How this after 7 days, after 14 days, how these cells are generated, this one maturation is carried out, and the secondary immune response and the tertiary, because of the mutation, these are the mutations are there. Because of these mutation, how the activation of the B cells is carried out. Now how this is working, how the natural B cells are there, how this antibodies, there the hyperaffinity of the B cell is increased. So only the production of the B cell is not sufficient for the reaction mechanism. The hyperaffinity is very important. Now what is the hyperaffinity? By example, this is, see for example, this is native B cells are there, antigen is attached to the B cell and it is migrated to the germinal center. So this is the activation, this much is the activation. Once activation takes place, the maximum numbers of the cells should be produced. So B cells with somatically mutated V genes, IgE with varying affinity of the antigen. So this is one, this is second, this is third. These are the different types of the somatically mutated hyperaffinited your B cells. They only B cells is producing affinity membrane follicle, these are preparing, and afterwards they are majority of this one B cells is there, high affinity B cells is there. These are the major maximum amount of the antibodies are produced, and these antibodies react with this bacterial cell, this is the antigen, and this is react with the antigen. Now how T dependent antigen is carried out, that is a antigen recognition, and the afterwards activation of the lymphocyte, and how the secretion is takes place. So this is a IgM and IgD B cells is there, the microbes is attached, they sensitize this cell B cell which produces the IgM and the IgD. Now through this helper cell stimuli is very important, that I told you basically the activation of the B cell is very important, and once activation takes place a clone is synthesized. This is clone expansion of the B cells, and once clone expansion takes place, differentiation of the clone leads into effector cell antibody secreted first in initial response, the IgM is synthesized, that is the antibody secretion start. Initially IgM antibody is start production, afterwards it is class switching, that IgM is switched to the IgG. Once IgM initially is synthesized, afterwards it goes to the IgG, and once this IgG is synthesized, then the actual defense mechanism start, that is called as a antigen antibody reaction mechanism. Simultaneously, the memory cells are generated. This is very important. Memory cell means when you infect the second time with the same antigen, that is called as a secondary immune response. No need to there will be a direct peak period, because immediately they start the production of the antibodies. Now, what is the anatomy of this one? The activation of the migration of T cells and T lymphocyte. This is a follicle, this is a lymph node, the structure of a lymph node, and these are the B cells are there. The bacteria is attached to this cell, and this is the activation. T-B cell interaction takes place. This is in lymph node. That is, one B cell it secretes this IgG isotopes secretion takes place. Then there is germinal center reaction, affinity maturation of the memory B cell. Because when there is a maturation of B cell, then only there is a production of the antibodies. So, in primary response, just see the peak of the primary response is very less, and if you see this is a lag period. But if you see in case of secondary immune response, there is no lag phase, direct peak is there. This is a secondary immune response. So here there is no recognition, because the memory cell already recognize what type of antigen is there. Here initially, to recognition of the antigen is very important. Unless there is a recognition, the IgM is not synthesized, then IgG is not synthesized. Now just see that I compare with the primary immune response with the secondary immune response. In IgG, the amount of IgG is produced in primary response is less as compared to the IgM, but in secondary response, IgM is very little amount of the IgM, the antibodies is produced, and high amount of the IgG is produced. Means basically in secondary immune response, IgG is produced in major one. Now antibody isotope switching. What is the meaning of the isotope switching? This is very important. This is activated B cell is there. This is activated B cell, that is a centroblast, or it is called as a blast. And once this blast is there, this proliferate into B cell, two B cells are there, this synthesize the plasma cell, which gives the IgG2A, IgG2, while this is synthesized IgA, and this is IgG, and this blast cell which produce the IgE, and this is IgG, and this is IgM. So there are different types of the cytokinins are synthesized by this cell, that is the activated cell, that is called as a IL-2, IL-4, IL-5, IFN, gamma, and TGF-B. These plays the important role in the defense mechanism. So how this isotope switching is carried out? Thus this is an example of recognition of B cell, microbial antigen, preparation of the DNA, no signal is there, and signaling helper cells are there. There is synthesis of the IgM antibody, IgG, and IgE. Now, how this antibody response is carried out? One is memory B cells is development in the secondary response, and second in the isotopic switching is there, affinity maturation is carried out. Now this is a comparison between a primary response and the secondary response. This is what is a secondary response and the primary response is shown. These are the T1 antigen and the TD antigen is shown. Now this is a thymus dependent antigen, where this is a nature is a protein, while the thymus independent, where this is a glycopeptide and the nucleic acid and the saccharides are there. Now how this role of helper cell, because the helper cell plays the important role in the cytokinin production, or CD40 and the CD40 interaction is carried out, while isotopic switching switch the recombination and the cytokinin and the isotopes. So antibody effector, what is this antibody effector? These effector has produced antibodies only react with the antigen, means what? The one thing is they carry out the neutralization of the toxin produced by the bacteria, that is called as a neutralizer. Second one, opsonization of the phagocytic microbes. Third is antibody dependent cytocytosis. Lysis of microbes are carried out. Phagocytosis of microbe opsonize with the complement fragment and the inflammation. Now even though this is true with the antibody and the without antibody, without antibody, how it happens, the bacterial cell comes inside and the degradation of the infected cell takes place. But if the antibody is there, how they protect other cells from the attack of the antigen. Now next, this is a pathological effect and the toxin producing by the bacteria, and that can be neutralized with the antibody. Antibody binds with this toxic and it will neutralize this one. And this is a opsonization of the microbes. How it is carried out, the opsonization by IgG. These are the opsonization, killing at the indigested microbes. And this is a natural killer cell. How plays the important role in the cells? That killing of the helminth. How helminth killing is worked by these cells. Now these are the opsonization. This is the best example of phagocytosis. Microbes are phagocytosed and destruction of the microbes by leukocyte is carried out. And this is the best photograph which is killed. This is a live bacterial cell. This is live bacteria and this is killed bacterial cell. And these are the photographs, and this is the immune response. In summary, this is a mechanism, innate immunity. That is a innate immunity. It is also called as herd immunity. It plays the important role in the defense mechanism, because rather than passive immunity, innate immunity is very important. This is a cell-mediated type of immunity. These are antibody-mediated immunity. Thank you.